So we're going to continue on taking a look at pathogenesis. And again, just to remind you, when we look at pathogenesis and you take that word and look at the roots there, you're looking at something that is going to create suffering in you. The genesis is the creation and the pathos is the suffering or emotion that you're going to be feeling. And when we look at these kinds of infections that are going on there, we really have to go back and consider the type of the organism that is causing the infection. And there's a wide variety of different types of pathogens that are out there that cause a variety of different kinds of diseases, but also we have to think about the condition of the host and how you can be immunocompromised just from stress or malnutrition, and that can lead to further advances in the pathogen causing the disease. And when we look at this, we do have to consider that we do have a normal complement of microbiota that are out there. The resident microbiota can actually be antagonistic to other cells because they're better adapted to living there. And a really good example of this is E. coli in your intestine is really adapted to living in those conditions, and they will tend to outcompete, but also cause other kinds of pathogens to not be very successful in what they're trying to do to cause these diseases. When we look at the pathogens that are out there, not only do you have the residents that are hanging out normally, but you also have the transients, and those transients are just passing through. So you consume something that has salmonella, and there is a chance that the salmonella bacteria can evade your immune system and also evade the E. coli in your intestines, and then they would be able to cause that infection. And we also have opportunistic pathogens that are out there that can cause a disease when the host is compromised. So this is something that you would really want to consider when you're looking at something like an immunocompromised patient that are out there. These pathogens are going to take that opportunity to cause the disease because they do have that ability. And when we look at the primary pathogen, these are ones that are going to be able to cause the disease regardless of the resident microbiota that are out there or your immune system. So there's, again, this wide variety in the different classifications of different types of pathogens that are out there. And if we look at the figure right here, what we can see is these different stages that we go through when we look at this idea of the creation of disease. And so we'll go through these step by step and spend a fair amount of time at looking at some of what's going on here, because again, this is what is going to give you your signs and your symptoms that are used to diagnose the disease and also allow us to define the syndromes that are out there. And so if we start out over here and we take a look at this first thing, the pathogen has to be able to get into the host cell that is, or the host organism that is out there. And when we look at this process, we can consider this idea of a portal of entry is how they're going to be able to get into the host out there, and they're going to start out with colonization. And when you look at colonization, this is really the presence of the microorganism that's in or on the host that is out there. They're going to be able to hopefully grow, and I shouldn't say hopefully, they're going to be able to grow and multiply, but you don't necessarily see any signs or symptoms there, and you don't necessarily see an immune response in the host. So you can imagine somebody spitting on you or coughing on you or sneezing on you. That can lead to the presence of that microorganism there. When that organism is able to infect, now there's a difference, because now we're looking at the successful colonization of the host, but now the microorganism is not only present, but it's multiplying. And this is where we need to be cautious when we look at the presence of pathogens, because if they can gain a foothold into your body and start to do this multiplication, then they're going to increase in numbers here. And so if we look at this first stage, it's really you're being exposed, and this organism is able to gain access to the host tissues through this portal of entry. And if you look at the different kinds of portals of entry that are out there, they're going to be able to get through your skin, often through a tear or a break in the skin to get access there. Really an easy method to, or I shouldn't say easy, an easier method to be able to get in is through mucous membranes that you're going to find in your nose and your mouth where you're breathing things in, but also when you're trying to expel waste products, you're going to have this mucous membrane there, this epidermal layer that has secretions of mucus to try and trap the pathogens that are trying to work their way in. You can also look at what are known as parenteral routes, which would be something where you've got a breach in your skin through something like a wound or a bite or even a needle in a doctor's office could cause a parenteral route of infection to allow entry into the tissue or into the host there. There is one that isn't really well summarized right here, but we can also consider a portal of entry is the placenta that allows pathogens to get access to a fetus. And this is where we look at what is known as torch infections that are out there. And when you look at these torch infections that are out there, it's sort of an acronym to give you some idea of some of the microorganisms that are going to be out there. You've got toxoplasma, which is a protozoan parasite that we talked about before. That is primarily the reason why they say pregnant women should not change kitty litter boxes, because they can get exposed to toxoplasma that can then get into the fetus and cause a variety of different types of damage. Interestingly, when you look at torch here, one of the other parts of the acronym there is other organisms. So syphilis, parvovirus, you can see varicella zoster virus, so your chickenpox, and also Zika virus was added to this list of others recently. We've got rubella, the MMR vaccination is going to be taking care of that. You've got cytomegalovirus, or CMV. That's a pretty nasty virus that actually causes some cells to fuse together, and so you get these monster cells, or cytomegalo. And then, of course, our herpes simplex virus that's out there. This does not rule out, there are a variety of other kinds of organisms out there, like Coxsackie virus, polio virus, and also Listeria monocytogenes is a nasty little bacteria that you can get from improperly treated deli meats that are out there. So they do recommend that women who are pregnant actually cook their deli meats if they're going to put them on a sandwich, because of the chances of Listeria. And it's a really interesting bacteria that will actually recruit cytoskeletal elements and build them up, and it'll use that to push the bacteria from one cell to the other. And so when this happens, it's actually going to cause the bacteria to work its way through the fetus there and cause a variety of different forms of birth defects. So now that we've seen that these organisms can get in through this portal of entry, we can go to the second stage here in pathogenesis, and this is where the pathogen is going to be basically attaching at that portal of entry using what are known as adhesins that are on the surface of the microbe that is out there. And when we look at these different kinds of adhesins, this is where we can see what are known as virulence factors. And depending on the type of virulence factor, you can have some organisms that are more likely to cause disease. And if you have an absence of those organisms will be less likely to cause a disease. And so if we look at these adhesins that are out there, again, we're just looking at something on the surface that allows it to attach. And we can see a variety of different strategies that these things have come up with, but also notice they're targeted to different tissues that you're going to find in these portals of entry. So this is going to allow certain microbes to be more successful going in through there. So you can see respiratory epithelial cells are where this protein F is going to attach for allowing this Streptococcus pyogenes to attach, and that's going to result in strep throat. You can see the organism that causes cavities here actually is able to attach to your teeth, and there you have Streptococcus mutans. We've seen with bacteria, they can use capsule layers. That glycocalyx is sticky. They can use fimbriae to attach, or sometimes they can even use flagella. And of course, we have our biofilms that are out there that can allow a whole bunch of these things to attach. When we look at protozoa, you're going to find that cilia or different kinds of hooks can be used to attach. And viruses, we've seen that those spikes on the exterior of the virion particle are going to allow them to be able to attach. So if you get rid of those spikes, the virus can no longer attach, and that's going to make it less likely to cause that disease. And so this is a good example why something like influenza or the COVID virus that have these envelopes with the spikes are sensitive to even just washing your hands with soap and water. Some of the other kind of interesting examples we can see here, when you look at the capsule around the outside of this bacteria, that could be used to actually cause it to be sticky, but they actually have found that depending on the structure of the capsule, sometimes this can be a virulence factor that allows it to go through the process of invasion, and there you're looking at not only have you successfully infected and you're multiplying, but now you're starting to spread throughout the local tissues or systemically throughout the body here. So you've attached and you're starting to replicate because you're basically inside of an incubator at this point. You've got food, you've got a nice warm environment, and sometimes that capsule can allow the pathogen to stick, but other times it can actually allow it to be more successful at surviving inside of the host. And in this little cartoon example, what we can see is normally this phagocytic cell is going to be patrolling, say it's in your lungs, and it's patrolling to find these bacteria cells that you have inhaled in, and they're in your upper respiratory tract. And it uses an antibody that is basically attached to this bacteria cell on these little yellow pegs here, the antigens, and this phagocytic cell has a receptor to bind to the antibody, and that effectively tethers this phagocytic cell to your pathogen, and then it's going to be able to consume it. Well, what they found is certain types of capsules out there actually are going to protect the bacteria cell and prevent the antibody from sticking, which means your phagocytic cell is less likely to be able to stick to the pathogen and consume it there. So this is, again, this idea of virulence factors. This is not only going to allow it to colonize, but also to invade and disseminate its daughter cells even further into the host that is out there. So this is an anti-phagocytic kind of mechanism for what we're looking here. Another sort of example that we can see when we look at these virulence factors here, we've got our capsule here, and not only can they avoid phagocytosis, but some of these bacteria can actually secrete protease enzymes that are specifically made to break down the antibody. So now, not only do you have the capsule, but you can break down the antibody, so it's less likely for the phagocytic cell to be able to eat you. If you have both of these virulence factors, you are more likely to cause a disease. We can see over here another interesting thing is this is blood plasma proteins, and certain bacteria can actually secrete different enzymes that will cause your plasma proteins to actually coagulate. So here we can see the blood plasma proteins are kind of liquid when you tilt it to the side, and in the upper tube you can actually see what happens as a result of the coagulase enzyme there. So imagine this bacteria is in your bloodstream. It's going to literally coat itself with your coagulated blood proteins, which means your body is not going to be able to recognize it because it looks like you. It's got your own coagulated blood proteins around the outside there. So really kind of an interesting way to hide out, and not all kinds of bacteria cells are going to have that coagulase protein available there. Over here in this last figure we can see what's known as the M protein, and this one is actually going to project outside of the peptidoglycan layer on our gram-positive cells, and this is kind of similar to what we see with capsules. This is going to block proteins that try and assemble on the outside of the cell to help your phagocytic cells to be able to recognize that pathogen there. So this M protein is almost like a little spike that prevents these complement proteins in your bloodstream from binding to the pathogen. We can also see things like the waxy mycolic acid coating around the outside of tuberculosis bacteria actually helps them to resist being digested when they are phagocytosed. So the phagocytic cell can eat it, but then they can actually resist the phagocytic mechanisms inside of those cells, so they're not going to die off. So a lot of different really interesting strategies when we look at these virulence factors. Here we can see something that is known as antigenic variation, and this is basically what cells can do is they can change the surface proteins around the outside here. So what the antibody would normally stick to allow the phagocytic cell to recognize it, the cells can actually mutate those things on a regular basis. And when we look at this, if you're looking at antigenic drift, this would be minor changes. So think point mutations in these H and N spikes around the outside of influenza virus. And every once in a while, you get what's known as an antigenic shift. So a drift is just a little change, but an antigenic shift, this would be where the virus picks up a completely different copy of this spike protein from another virus that's out there. And your virion particle now has a totally different complement of these things around the outside. So you're not going to be able to recognize this thing with your antibodies, and it's going to be able to more successfully invade your body there and also hide away from your immune system response. So this is a really nasty kind of thing that we do see with influenza virus that is going to cause something like an epidemic or a pandemic. When you do see these recombined versions of viruses. And when we look at influenza virus, it can actually cause infections in birds and mammals, so it gets transferred back and forth. If you remember the picture of the little kid making out with the pig there, that can be how you get that zoonotic disease transmitted from a pig into us. And if the pig had influenza A and the kid had influenza A, now that kid could make one of these antigenic shift versions where they've combined some of that pig version or the swine flu version of influenza. So now that this pathogen has gotten into the host and they're surviving that, now we can start to see some of the disease signs and symptoms that are being caused by this, because oftentimes these pathogens will cause damage to the host there, and that's what's going to be producing these things that can be measured or that are being experienced by the person that's out there. And again, this goes back to that idea of the virulence factors that can help the pathogen to invade further and cause damage to the host there. And when we look at these virulence factors, they come in a variety of different forms here. One type is a secreted form that's known as an exoenzyme. And we sort of looked at exoenzymes in the lab when we looked at their ability to consume different kinds of polymers. Well, here you can see different examples of these different kinds of proteins that are out there. You can see nucleases and proteases. You can break down membranes, but you can also break down the molecule that's used to hold the cells together. And if you think about that, that's going to allow this pathogen to work its way through tissues, and that will allow it to disseminate even further. So really, when we look at this exoenzymes, these are literally going to be eating the host cells and tissues to spread further throughout your body there. Another example of these kinds of things are what are known as toxin molecules, and they come in two flavors. One of them is an exotoxin that is actually going to exit the cell, and this one over here is an endotoxin, which is literally a part of the cell. And we can see some of the characteristic features of these that we'll go through, because you can see there is some distinct differences between these two classes of toxin virulence factors. The first thing that we can look at is, where do these come from? The exotoxins, like I said, these are secreted from cells. We can find them with gram-positive and gram-negative cells, and also with fungi and protozoan cells can secrete these exotoxins. With endotoxins, this is a feature of the gram-negative cells, which are very different from the others because they have that outer membrane layer, and so the endotoxin is literally a part of that outer membrane, and it's going to get released when these cells are dying. If we look at the composition of it, your exotoxins are proteins that are made and secreted by these cells that are out there, whereas your endotoxin is that lipopolysaccharide. Remember, your lipid A from the outer membrane of these cells that are out there. If we look at the effect on the hosts that are out there, the exotoxins are the ones that are going to cause specific damage to your cells that are out there. And so if you look at something like cholera, it's going to actually cause an imbalance in electrolytes in your lower intestines that's going to result in things like diarrhea. We can also see nausea. If you look at the staphylococcus toxins that are out there, those can result in vomiting. So we see nausea, but oftentimes when we look at exotoxins, these are going to cause death. So they are much, much more hazardous there. And it's not to say that endotoxins are bad, but these guys are typically going to have systematic effects like inflammation, and you will also see a fever. So while these things are going to cause direct death of cells and tissues, these are going to spread throughout your body and have an effect on the whole organism that are out there. If we look at an interesting feature is heat stability. And if you think about this, you are generating a fever to try and reduce the effects of the pathogen. But notice, the organisms that are going to result in fevers, well, their particular toxin is heat stable. That LPS, or lipopolysaccharide, is not sensitive to heat. I should say body heat, as opposed to your proteins that are out there that are more sensitive. So most of your exotoxins are sensitive to fevers, and that's going to help to denature those proteins. So this does provide a little bit of a protection where the LPS is heat stable and you develop that fever and it does not go down. And if we look at this, the other interesting feature is antitoxins that are out there. These would be things that we could administer with antibodies in them to block the effects of the toxins. Well, proteins, there are antitoxins available for these, but LPS tends to be a little bit slippery from that lipid portion, so we can't administer something to directly block the effects of that LPS. So you're going to, again, get that inflammation and that fever that is going to develop, and it's going to keep going on and on and on. Luckily, when we look at this, the lethal dose that kills off 50% of hosts, or the median lethal dose that's out there for exotoxins, this is extremely low. So think Botox that's out there. With endotoxins, you do need to have a higher dose in order for it to kill you, but remember, these are heat stable. And when you finally get around to identifying and treating the pathogen, the more that are there, the more LPS is going to be released when those pathogens are killed. And so that's more likely to result in death of the patient that's out there. The textbook does go through, and you can see I'm not too concerned about all the details in these, but I just want to show you that there are three main mechanisms for these exotoxins to work. One of them is going to target between the cells that are out there. The second one, we can see these guys actually are specifically made to disrupt membranes in cells. And then the last one that we can see is what are known as superantigens. And these are some nasty little things that actually take your normal immune system response and cause the release of what's known as a cytokine storm. So you start releasing these chemicals that are normally part of your immune system response, but then you release so many of these that your immune system response just goes crazy. And the worst kind of thing when you look at some of this stuff is it can actually cause you to go into shock that can result in death of the patient. So these are some of the really nasty kinds of things that we can find associated with certain types of pathogens that are out there. If we look at different kinds of organisms that are out there, things like fungi and the helminths and the protozoans, you will find that they all do share some different kinds of virulence factors that are out there to allow these kinds of pathogens to be able to survive a little bit more effectively in the hosts that are out there. Step 5, we can take a look at this idea of the transmission of the disease. Now that you've gotten in, you've successfully invaded and spread around, you've caused damage, you've been able to replicate, and you've made some new particles, whether they're viruses or cells. Now you need to get out, and this is where we see the opposite of our portals of entry, our portals of exit. Again, through your skin, oftentimes through the respiratory tract because you cough and you sneeze, but also we can find them coming out through those mucous membrane sites like your urogenital tract and your gastrointestinal tract.